Dr. Sandeep Kumar Mishra and I am discussing the various aspects of pharmaceutical microbiology. So, what is aseptic area? Microorganisms are present all over the environment, but sometimes these are pathogenic in nature, and there are certain areas which needs complete removal of the microorganisms, like the places where surgeries are performed, or the places where the drugs or the important products which are used for the consumption of the human beings are formed. So for the production of these areas, for the production of these products, some specific area is required, which is called as the aseptic area. So what is aseptic area? Aseptic area is an area which is completely free from the microbes. So here there is complete check for the growth of the microorganisms. So the entire production area, which includes also the aseptic area, consists of various components. The first is the cleanup area, the second is the compounding area, the third is the aseptic area, the fourth is the quarantine area, and the fifth is the packaging or the labeling area. So what is cleanup area? Cleanup area is that area where the incoming raw products are cleaned before actually keeping into the compounding area. So what is compounding area? Compounding area is an area in the production house where the formulation is made or where is compounding is done. And then there is aseptic area where finally all the chemicals are processed and kept in such a way that they can be protected from the environmental microorganisms. And then there is the quarantine area. So what is quarantine area? Quarantine area is an area in which the produced or manufactured products are kept before packaging or before transport. So in the quarantine area, the manufactured products are kept for the short duration of time before transporting, and then finally the packaging and the labeling area. So for producing the aseptic environment in the laboratory, one of the most important equipment used is the laminar airflow. So what is laminar airflow? Laminar airflow, it is an equipment which gives a laminar flow of air so that the external air cannot enter inside the aseptic area and cause the growth of the microorganisms. So the laminar flow equipment is basically of two types: vertical laminar airflow and the horizontal laminar flow. So in the vertical laminar flow, the laminar air comes from the top and it moves to the downwards. It is usually, the vertical laminar flow equipment is usually kept in the doors, so that when the entry of the humans or the personals which are working in the area occurs, so that during that time the microbes cannot enter in the room. And then the second type of laminar airflow is the horizontal laminar airflow bench. So this type of laminar airflow is usually present in the laboratories where microbes or other cells are cultured, and this type of laminar airflow bench is used for performing various type of cell culture or the microbial activity, so that the microbes or the cells cannot get contaminated by the other organisms. So if we talk about the sources of contamination in aseptic area and what are the methods for the prevention of the prevention of contamination, then basically there are three major sources for the contamination. First is the atmospheric contamination, second is the fluid contamination, and then third is the transfer contamination. So what is the atmospheric contamination? So atmospheric contamination is the contamination which takes place due to the environmental condition. So what this means is that if the walls or the manufacturing area is contaminated with the microorganisms, then there are chances that these microorganisms gets entered in our final formulation and contaminate our formulation. So one of the most important source of microbial contamination in the formulations is the atmospheric contamination. Then how you prevent the environmental or atmospheric contamination? First is the use of the electrostatic precipitator or the HEPA filters, so that the air which is entering into the room is completely free from the microbes. And the second most important method is the complete or periodic removal of the air from that area, or the dust which is settled on the walls, floors or ceilings. So this is the way by which we can prevent the contamination, atmospheric contamination. And then the second type of contamination is the fluid contamination. So how does fluid contamination occurs? So in the pharmaceutical industry, a lot of fluids are used, and one of the most important or the greatest solvent which is used in the pharmaceutical industry is water. So there are chances, if the water is contaminated with microorganisms, then there are chances this water also spoils your final pharmaceutical product. So if the water is not pure or the water is containing microorganisms, then it might finally affect your formulation. As you know that sterile preparations or like sterile water for injections, or usually the formulations which are directly injected into the veins, should be completely free from the microorganisms. Hence, the preparation of such formulations requires complete removal of microorganisms from the solvent. Hence, one of the major source of the fluid contamination is water. And then the third type of contamination is the transfer contamination. So what is transfer contamination? Transfer contamination is that type of contamination which occurs when the final product which is formed is transferred from the one place to another. So if there are microorganisms in the if there are microorganisms present in the environment, then it might be possible that atmospheric dust or microbes gets incorporated into the final product during contamination. So this process is responsible for the contamination in the final product. So hence the transfer contaminants occurs during transportation process. So how you can prevent transfer contamination? So transfer contamination can be prevented if we use face mask or head cap or gloves and food covers, and personals which are responsible for the transport of the products are well trained and periodically evaluated in the principles of aseptic processing, and they should be taught with the techniques how the entry of the microbes or dust particles can be prevented inside the final product. So these are the different methods, and one of the best method to determine the contamination is to periodically assess the air which is present in the production area. Now, how you will determine the concentration of the various types of products? There are certain products which for measuring the concentration, the chemical methods are not available. So there are certain products whose concentration or strength can be determined using the assay with the help of microorganisms. So such types of assay are called as the microbiological assay. So what do you mean by microbiological or biological assay? So microbiological or biological assay refers to the measurement of relative potency or activity of the compound using microorganisms. Hence, it is the type of biological assay employed on microorganisms for the determination of relative potency of activity of any microbiological compound. So basically, antibiotics, vitamins, and amino acids can be quantified or assayed with the help of microbiological assay. So one of the best method for assessing the potency of the antibiotics is the cylindrical plate method. In the cylindrical plate method, small cylindricals are poured into your petri dish containing solidified media, or we can say agar media, and then your antibiotic solution is poured into the cylinders, and then the zone of inhibition which is formed is evaluated. So the greater is the length or diameter of the zone of inhibition, the greater is the potency of the antibiotics. And then finally, what is spoilage? So spoilage is the term that indicates the goods that do not meet the production standard. So it is the method by which material or the amount of material is wasted or scrapped during the manufacturing process. So in another word, it is a form of substandard drug or food in which the quality or value or even the usefulness of the article is destroyed or impaired by the action of microorganisms like fungi or bacteria, so as to render the article unfit for human consumption. So if we talk about the types of spoilage, then the spoilage is basically of two types. First is the microbial spoilage, and second is the non-microbial spoilage. So if we talk spoilage, spoilage is basically divided into two parts, microbial spoilage and non-microbial spoilage. And the non-microbial spoilage is further of different types like chemical spoilage, enzymatic spoilage, or physical spoilage, or there may be other types of spoilage. So what happens in chemical spoilage? So chemical spoilage is the type of spoilage in which actually the chemical composition of the major ingredient gets converted into some other form. Hence the final or the main ingredient which is present in the formulation, its chemical composition or chemical strength gets changed. Then there is enzymatic spoilage. In this type of spoilage, usually enzymes are responsible for producing the spoilage, like the sourness or bitterness of the milk occurs due to the enzymatic spoilage. And then there is physical spoilage. So what is physical spoilage? In the physical spoilage actually involves the changes in the physical state of the formulation. So usually physical spoilage we can see in case of emulsions, where if the emulsifying agents gets spoiled, then your final emulsion gets destroyed or spoiled as the different oil and the water phases get separated. So these are the different types of spoilage. So what are the various factors that affect the microbial spoilage of pharmaceutical products? So we can say that these includes the types and size of contaminant inoculum, or the nutritional factors, or the water which is the solvent which is used. If it is impure, then it is responsible for the spoilage of the final formulation. And then the storage temperature. So every product is stored at specific temperature. It has specific storage conditions. So if that product is not stored at that particular temperature, then there are chances that the products get spoiled or spoilage is there. And the pH is also very important factor which prevents the spoilage. So if there is no optimum pH, then there are chances that the final products get spoiled. Then oxidation or reduction balance. So there is proper balance in the redox potential of the formulation. If it is oxidized or reduced, then there are chances of a spoilage. And when finally the package design. So package design should be such that it does not allow the foreign or the atmospheric contamination or microorganisms to enter into the final formulation. If the formulation gets in contact with the microbes or dust, then there are chances that the final products get spoiled. So then finally, how you will preserve your formulation through the spoilage. So one of the best methods for preventing the spoilage is the use of the preservatives. So what are the different types of preservatives? So preservatives can be cationic detergents or alcohols or phenolic compounds or organic acids. So what are the various factors that affects the efficacy or availability of the preservatives? So one of the factors which is affecting the efficacy of preservative is the temperature, or it may be the chemical structure of the preservatives, capacity of preservatives, inoculum, effect of pH, effect of containers and packaging, and then changes of concentration. So how these preservatives work? So these preservatives work by three major mechanisms. First is the antioxidant mechanism, second is the antimicrobial mechanism, and third is by chelation. So one of the best method for preservative mechanism of action is the antioxidants. So these are the products which, when kept into the formulation, they prevent the oxidation or the oxidative degradation of the final product. So various antioxidant preservatives which are used are vitamin E or vitamin C. So these are the products which, if kept into the formulation, they prevent the oxidation of your formulation. And then second is the antimicrobial agents. So some formulation gets spoiled if some microbial contamination is there in that formulation. So your formulation, if it contains antimicrobial agents, it prevents the growth of the microorganisms in that formulation and hence prevent the spoilage. So one of the best method to prevent the growth of the microbes in your formulation is the use of the antimicrobial agents. So various antimicrobial agents are available. Among these, the major important ones are the benzoates or sodium benzoates or sorbates. And then finally the chelating agents. So what are the chelating agents? Chelating agents are nothing but the complex forming agents. These agents form the complex with the environmental pollutants or heavy metals which are present in the environment and enters into your final pharmaceutical products. So when chelating agents is present in your formulation, these chelating agents forms complex with the either heavy metals or the external contaminants, and then these entrap these contaminants into themselves. Hence, with the help of these chelating agents, your final products gets prevented from the contamination. And then finally, what is animal culture? So this is the culture which is used for studying the various types of mechanisms of your drug action, or the mechanism of how various types of phenomena occurs in the microbes or in different types of cells. So the cell culture or the animal cell culture may be of two types. First is the primary culture or the secondary culture. The primary culture basically involves the direct culture of the cells from the tissue or the organ, and then the secondary culture is the culture which is obtained by passaging of the primary culture. So cell culture or animal cell culture is very important, as it is one of the major methods by which we can prevent the direct inoculation of the compounds or the drugs needs for testing into the animals. Hence, we can test the drugs or antimicrobial agents in the culture media, and then we can finally extrapolate our results from the microorganisms to the final humans. So these are the various aspects of the microbiology which I want to discuss.